Hello everyone, welcome to the Bodhi Academy. In today's video, we are going to continue with 12th Standard Chapter 11 Biotechnology Principles and Processes. So, in the last class, we had learned about the gel electrophoresis process, which is used to separate the different strands of DNA. So, today we will learn about the cloning vectors. So, let us begin. Now first of all, what are cloning vectors or what is a vector? So a vector is a DNA molecule which is used to introduce the foreign DNA or the recombinant DNA which we have cut into a host cell. Okay, so finally whatever DNA we have cut and the isolated, we need to transfer it to a host cell where it can replicate. So a vector is a DNA molecule used to introduce the foreign DNA into a host cell. It is capable of self-replicate and integrating into the host cell. So a vector is capable of self-replicating in the host cell and integrating itself into the host cell. Then vectors can be plasmid from bacterium or a cell from a higher organism, or it could be also DNA from a virus. Now the target DNA is integrated into the particular sites of vector and ligated using the DNA ligase enzyme. So this is the enzyme which is used to link or join different DNA molecules, and the target DNA, whatever isolated DNA that we have got, it is integrated in the particular sites of the vector DNA and they are joined using the DNA ligase. Now the host cell is subsequently modified to receive the vector, allowing the replication. Then the DNA molecule, which when a foreign DNA is inserted, has the ability to replicate autonomously to give birth to multiple clones of recombinant DNA, is called as a cloning vector. So what is a cloning vector? It is a DNA molecule which, when a foreign DNA is inserted into it, has the ability to replicate autonomously, means by itself, or self-replicate to give birth to multiple clones or copies of the recombinant DNA. Then again, as we said, plasmids and phages are cloning vectors, are examples of cloning vectors. Then talking about the characteristics of cloning vector. What are the characteristics or the features that a cloning vector should have? So the first one is origin of replication or ORI. So what is origin of replication? It is the specific sequence of nucleotides in DNA which serves as the origin of replication process is called, and it is called as the ORI. Now when it integrates or binds to the sequence of foreign DNA, the DNA starts replicating alongside the DNA host. So as we can see here in this diagram, this part is the ORI or the origin of replication of this vector. This is a E. coli cloning vector, and it is the specific sequence of nucleotides in the DNA which serves as the origin of replication. So from here the replication will start. Then the second thing which is required is selectable marker gene. So the cloning vector must contain a selectable marker gene, as this enables the selection of the host cell which contain the recombinant DNA. So it will help in identifying those host cell which contain the recombinant DNA, that is the rDNA, and the separation of them from those host cell which do not contain the rDNA. Then another thing which is required is the presence of restriction site. Now it should consist of restriction sites to allow the breakup of specific sequences with regard to the restriction endonucleases. So with regard to the restriction endonucleases or restriction enzyme, there should be restriction sites which will allow the breakup of the specific sequences. Then another thing is that the vector DNA, it should not be too big in size. Okay. Then there should also be several sites for cloning. So multiple cloning sites should be present. Multiple cloning sites should be there in the vector DNA. So as we can see here, now these are all the restriction sites. We have E. coli into BAMH1 cell, then these two. So all these are the restriction sites. Then there is the origin of replication, ori, and the antibiotic resistance genes, which are these two. So this is the features or characteristics of a cloning vector. Then talking about the competent host. Now this competent host is required for the transformation of recombinant DNA. So how this recombinant DNA or how these vectors are introduced in the competent host. Now first of all, the first method is simple chemical treatment with divalent calcium ions increase the efficiency of the host cell to take up the recombinant DNA plasmids. Then another method is the recombinant DNA can also be transformed into host cell by incubating both the recombinant DNA as well as the host cell on the ice, first on the ice, followed by placing them briefly at the heat shock, that is 42 degrees Celsius, and then putting them back on the ice. Now this change in temperature will help the bacteria to take up the recombinant DNA. Then another method is the microinjection method. So next one is the microinjection method, where the recombinant DNA is directly injected into the nucleus of the cell. So here the recombinant DNA is directly inserted into the nucleus of the cell, and this is done using a micropipette. Then next method is biolistics or the gene gun method, and it has been developed to introduce the recombinant DNA into mainly the plant cell using a gene or particle gun. Now in this method, the microscopic particles of either gold or tungsten are coated with the DNA of interest, that is the recombinant DNA, and they are bombarded into the cells. Then the last method uses the disarmed pathogens. So in this, the pathogens are disarmed and they are used as vectors. So they are weakened, and example is Agrobacterium tumefaciens, which when allowed to infect the cell, they will transform the recombinant DNA into the host cell. So this is how the transformation of recombinant DNA into host cell or competent host is done. This is all in this video. Hope you have understood this. Thank you for watching my video. Please like, share and subscribe to the Bodhi Academy.